introduce you to the group. So Jeremy is a second-year postdoctoral fellow with a broad interest in understanding the mechanisms that regulate skeletal muscle size and functioning in health and disease. His PhD studies at the University of New Mexico cover diverse areas, including exercise physiology, exercise immunology, and muscle physiology and pathology. Currently is under the co-mentorship of Dr. Andrew Judge and Dr. Sarah Judge at the University of Florida. And through the support of an NCI-funded team-based interdisciplinary cancer research training program, his research investigates the regulation of muscle mass and cancer-associated muscle pathology in preclinical models of cancer-induced cachexia, both with and without chemotherapy, to uncover novel mechanisms and therapeutic targets in cancer cachexia. I would like to give the floor to you, and I'm happy to listen to your talk.
All right, thank you for that introduction. So yes, my name is Jeremy Ducharme. I'm a postdoc at the University of Florida, working in the Judge Lab under the mentorship of Andy and Sarah. And I'm really excited to talk to you all today. And I want to thank the Education Committee for the opportunity to share some of our work on pancreatic cancer in the skeletal muscle clock. So as this group's well aware of, the vast majority of patients with pancreatic cancer present with cachexia, a devastating metabolic condition characterized by significant loss of body weight due to loss of muscle mass, with or without the loss of fat mass. And unfortunately, cachexia ultimately results in drastic reductions in quality of life, treatment tolerance, and survival in these patients. And it's in this regard that we in the Judge Lab have been studying mechanisms underlying the initiation and progression of muscle wasting in response to tumor burden, with specific focus on the FOXO signaling pathway, as we've shown that FOXO family members are increased in skeletal muscle in response to tumor burden, as shown here with the increase in FOXO1 and FOXO3A in the soleus and tibialis anterior muscles, as well as we've shown that the FOXO-dependent transcription is necessary for muscle wasting induced by tumor burden. And this led us to search for what's a FOXO-dependent target that is perhaps mediating these effects. And through doing transcriptome-wide analysis of tumor-bearing mice with and without intact FOXO transcription, we identified the transcriptional factor FOXP1, which we found to be increased in cachectic skeletal muscle in both pancreatic cancer patients, as well as multiple preclinical models of cancer cachexia. And it was associated with cachexia in patients with pancreatic cancer, as shown here with the increase in body weight loss and reduction in skeletal muscle index with increasing FOXP1 mRNA expression in skeletal muscle. We further found that inducible skeletal muscle specific FOXP1 upregulation is sufficient to cause skeletal muscle wasting and weakness, as reflected here with the leftward shift in fiber size cross-sectional area in the tibialis anterior muscle when we overexpress FOXP1, as well as this H&E image, which shows that when we overexpress FOXP1 in skeletal muscle, we get morphological and pathological changes to the soleus muscle that recapitulate what we see in cachectic muscle, as well as reductions in specific twitch force and tectonic force with the overexpression of FOXP1. And then recently, we showed that skeletal muscle specific knockout of FOXP1 protects against muscle wasting and weakness in an orthotopic model of pancreatic cancer cachexia. However, the FOXP1 direct targets and its transcriptional network in skeletal muscle remains largely unknown. So to address this, we performed FOXP1 chip sequencing in skeletal muscle of cancer-free mice and mice with pancreatic cancer at cachexia onset, which in our hands in the orthotopic KPC model occurs 12 days after injection, as reflected by reduction in body weight and reduction in skeletal muscle mass. And this FOXP1 chip sequencing implicated FOXP1 regulation of circadian rhythm. Specifically, we found that the core clock activator BMAL1 emerged as a top-ranking de novo motif within FOXP1 bound DNA regions in both skeletal muscle of cancer-free mice, as well as muscle of mice with pancreatic cancer. We further found that of genes with FOXP1 peaks shared by both conditions, circadian rhythm emerged as the top-ranking pathway, And this included core clock components such as the core clock activator BMAL1, as well as core clock repressors, PAIR1, CRY1, and CRY2. So a little background on the circadian clock system, and I will admit this is a gross oversimplification, but it is a transcription translation feedback loop that anticipates and regulates biological processes to maintain homeostasis. And it exists in nearly every mammalian cell. So on the left, we see this diagram where we see BMAL1 and clock forming a heterodimer that then is going to drive the transcription of core clock components that regulate BMAL1 and clock expression, as well as drive clock outputs or rhythmically expressed genes that are then going to go on to regulate rhythmic biological processes. So we wanted to find out, well, does pancreatic cancer influence the expression of core clock components? And is that mediated by FOXP1? So to do this, we took skeletal muscle from wild-type mice and mice with FOXP1 skeletal muscle knockout with and without pancreatic cancer. And then we performed PCR on select core clock genes that we identified as having FOXP1 peaks in their promoter region, as well as other core clock components. And we found that cancer-induced FOXP1 causes substantial disruption to the gene expression of these core clock components, including the repression of core clock activators BMAL1 and clock, as well as the upregulation of core clock repressors, including the period and cryptochrome genes. And I want to pause here just to highlight the potential significance of this cancer-induced repression of skeletal muscle BMAL1, as others have shown that muscle-specific BMAL1 deletion has been shown to cause muscle metabolic dysfunction, muscle atrophy, and muscle weakness, all of which are phenotypes common to cancer cachexia. Moreover, the function of the circadian clock is primarily to regulate metabolism, which in muscle, this includes glycolytic metabolism peaking during the active or feeding phase, and oxidative metabolism peaking during the rest or fasting phase. And since the circadian clock mechanism is a way of regulating predictive homeostasis, in order for these processes to peak during these times, the transcription or the upregulation of their genes would then need to happen near the transitions. So in this case, you would see genes involved in oxidative metabolism peak near the transition to rest, and genes involved in glycolytic metabolism peak during the transition to the active phase. So we wanted to test whether pancreatic cancer is influencing circadian patterns of gene expression, and whether that influence is mediated by FOXP1, and if these processes are disrupted. So to do this, we performed a circadian time course experiment in which we orthotopically injected PBS, or pancreatic KPC tumor cells, into the pancreas of wild-type or FOXP1 skeletal muscle knockout mice. And then beginning at the onset of cachexia, and every 4 hours after, we collected tibialis anterior muscles, performed RNA sequencing, and then we leveraged a rhythmicity detection package to identify the circadian transcriptome. By extracting core clock genes from this transcriptome, we found that core clock genes remain rhythmic, but cancer disrupts the circadian patterns of the clock's negative arm in a FOXP1 dependent manner, as I've highlighted here with these red boxes, with the wild-type response in the top row and the FOXP1 skeletal muscle knockout in the bottom row. Looking at the entire skeletal muscle circadian transcriptome, where here each dot represents the peak time of expression of a single rhythmically expressed gene, we see that in control muscle, these peaks occur evenly throughout the day, likely reflecting time of day dependent biological processes. And I've highlighted here, of these processes, we see that genes involved in lipid metabolism and carbohydrate metabolism display rhythmicity and peak in alignment with the natural rest-wake cycles that we just talked about near the transitions between active and rest. In response to pancreatic cancer, there's a pathological clustering of these rhythmic responses near the transitions between active and rest, where genes involved in metabolic processes are no longer rhythmic, but instead replaced by genes involved in inflammation and muscle catabolism. However, when we knock out FOXP1 from skeletal muscle, not only do we maintain the even distribution of rhythmically expressed genes around 24 hours, but genes involved in lipid metabolism and carbohydrate metabolism continue to display rhythmicity and peak in alignment with their natural rest-wake cycles. We then look to see whether there was time of day dependent effects on differentially expressed genes by looking at each of our 6 circadian time points again with the wild type response on top and the FOXP1 knockout response on the bottom. We found that cancer does cause time-dependent effects on these differentially expressed genes. And in wild-type mice, the greatest number of differentially expressed genes occurred near the transitions between phases. And this included a substantial downregulation of genes involved in oxidative phosphorylation following the transition to rest, which we just discussed. Genes should be peaking and upregulating at this time. And on the other side, we see that genes were involved in pathways classically known for muscle atrophy are peaking and upregulated following the transition to the active phase. And it's important to note here that for the majority of these differentially expressed genes, the dysregulation of these pathways required FOXP1. So far, skeletal muscle specific FOXP1 deletion we've shown protects against cancer-induced muscle wasting and weakness, prevents cancer-induced disruption of clock gene expression, and normalizes circadian patterns of gene expression in response to cancer. And that skeletal muscle specific FOXP1 upregulation is sufficient to cause muscle wasting and weakness. But is it sufficient to dysregulate core clock and clock control genes? So to address this, we performed RNA sequencing on skeletal muscle from mice with inducible skeletal muscle specific FOXP1 overexpression, and then extracted core clock genes that we highlighted earlier, again with and without FOXP1 peaks in their promoter region. And we found that this FOXP1 upregulation is sufficient to dysregulate core clock components, including the downregulation of core clock activators, BMAL1 and clock, and again, an upregulation of core clock repressors, including the period and cryptochrome genes again. In further support of FOXP1 as a negative regulator of clock function, we overlap genes differentially expressed by FOXP1 overexpression to genes differentially expressed due to a loss of clock function due to BMAL1 skeletal muscle specific deletion. And we found that of the 1,739 genes differentially expressed by FOXP1 overexpression, 405 of those genes were also differentially expressed in BMAL1 skeletal muscle knockout mice. And when considering the function of these genes, we found that genes commonly disrupted by both conditions are linked to pathways that have been shown to be altered in cancer cachexia. This included genes involved in beta oxidation, protein degradation, carbohydrate metabolism, as well as muscle structure and function. So our findings thus far, we see that circadian patterns of gene expression are disrupted in response to cancer, and that this may be mediated, at least in part, via FOXP1 dependent upregulation of clock transcriptional repressors. But are these findings translationally relevant? To address this part, we leveraged our previously published transcriptomic dataset from skeletal muscle of patients with pancreatic cancer and extracted core clock repressors and found that these clock transcriptional repressors are increased in skeletal muscle of cachectic patients with pancreatic cancer, and that they correlate with FOXP1 expression. We further found that these clock transcriptional repressors are negatively associated with skeletal muscle mass in patients with pancreatic cancer, in support of our preclinical findings. So to summarize what we've talked about today, cancer causes a loss of rhythmicity in circadian programs and muscle that regulate metabolism. Inflammatory and catabolic programs gain rhythmicity in muscle in response to cancer. And that skeletal muscle FOXP1 is required for cancer to induce circadian disruptions in muscle. And that direct targeting of core clock components by cancer-induced FOXP1 may contribute to cachexia. And with that, I'd like to acknowledge all those that helped with this project, our funding sources, your time, and I really want to highlight the consistent support and mentorship of Andy and Sarah, for which this wouldn't be possible. And with that, I'm open for questions.
Thank you, Jeremy, also for your great talk. It was really interesting to listen to it and to hear about the circadian rhythm. I would like to invite everyone to put their questions in the Q&A, if you have any. So I do have a question because you started indeed with the more preclinical work and then you ended your talk with translating it to more clinical work. So I was thinking about what would be your, so you have done already a little bit of clinical, translated it a bit to the clinical side of the spectrum. What will be your next step in this regard? Because I think indeed it's important, and a lot of clinical work is also shifting to the involvement of the circadian rhythm related to exercise and
Yeah.
cancer and chemotherapy as well. So just happy to hear your thoughts on this.
Yeah, I think there's still more work in the space in the preclinical, of course, but
Mhm.
I'd like to ask more perhaps translationally relevant questions of, there's other problems that we see with our patients in terms of the circadian disruption. Sleep is a huge,
Yeah.
a lot of cancer patients have disrupted sleep. So looking at doing studies with sleep disruption, um, different like timing of chemotherapy, timing of treatment, kind of the chronotherapeutic approach. There's a lot of interest there. So I think perhaps looking at what's disrupted in terms of a patient standpoint and then modeling that in our preclinical models is kind of the next step for where we're at.
Yes, thank you. I see a couple of hands, one from Lar. Can you, yes, unmute yourself?
Thank you
Yes.
very much, Jeremy, for your talk. I may have missed it, but I'm wondering to what extent you can place your finding within the broad spectrum of the multi-tissue organ crosstalk, because you're probably aware of the work of Mauricio Barrial-Diaz and his team, where they focus on the alteration of the circadian rhythm in the liver rather than in the muscle. I'm wondering, what is your perspective on that, or do you think this connects together?
Yeah, I think they pair each other really well. And I think I saw him in the audience, so it was cool to see him listen to this. Hopefully he felt kindly about it. Um, but yeah, I think the circadian disruption, I think there's more work to be done, of course, the spectrum of it in tissues throughout a cachectic individual, a cachectic mouse host. It's far from well understood, but I think there's many different levels of circadian disruption. I think we're seeing this in the tibialis anterior, so locomotor muscle, the liver, seeing that's been well established. Um, the crosstalk of whether that circadian disruption is then driving the circadian disruption in the TA. That's a really interesting thought. Um, I'm not exactly sure. I know there's a lot of interest in inflammation as a regulator of circadian disruption and then circadian disruption as a regulator of inflammation. So it's hard to know which is happening first. It's hard to think of if perhaps the circadian disruption in the liver is then driving a more inflammatory environment that then is contributing to the TA disruption. So it'd be interesting to look at other tissues and then compare what's happening in our model and kind of build more evidence of what's contributing where.
Yeah, great. Thank you very much for the talk again.
Thank you.
Thank you. Then I would see one more hand. Fabrizio, can you
Oh,
also?
yeah, thank you so much. Great talk. I don't know if I missed, but do you, in your system, this regulation of the clock gene expression is following or precede the onset of muscle atrophy or muscle alteration in cancer cachexia?
So that's one thing that we have more work to do on because all of these time points, this is at the onset of cachexia in our model. So we don't necessarily know what's happening with the disruption to the clock as we progress into worsening cachexia. But we also don't necessarily know when is this starting? Is this preceding cachexia? So there's more work that we need to do to think about whether either side of the spectrum of progression or perhaps a early event of circadian disruption. But everything I presented today was at the onset of cachexia.
Thank you. Let's see. Then we have one final question because we're running a little bit out of time, but I think it's good to answer the last question. So do you know if mice naturally exhibit circadian disruption due to tumor settings, and if manual controlling adds on to it? And does it correlate to level of disruption in patients?
So in terms of is the circadian disruption, in terms of the muscle, there's really not a lot, actually, in terms of anything peripheral to the tumor, there's really not a lot of work on circadian disruptions caused by cancer. There's a decent amount of work in the tumor itself of being dysregulated. So I don't know necessarily if like, if the mice naturally exhibit circadian disruption due to the tumor. I don't know. We're building evidence to support that may be just part of the pathology of cachexia, is that we're getting this circadian disruption due to the tumor or other factors released from the host. I'm not quite sure I understand the question of if manually controlling adds on to it. And if it correlates to the level of disruption in patients. We have some evidence at a single time point that the expression of these clock genes are disrupted, whether the circadian patterns of gene expression in the patients is disrupted and to what extent. We need to do more work on that.
Great, thank you very much. And since we are over 7 o'clock, I would like to finalize this nice webinar series of today. I would like to thank all speakers, Jeremy, Laura, and Nikita. It was really great to hear the work that you are doing, and I hope you could keep continuing the great work you are doing. We have a next trainee talk session in September, and of course another webinar series next month with Professor Laird and Kristin Solheim. So I hope to see you all there again, and for now I want to wish you a good night or a good afternoon for those at the overseas. Thank you all and have a good day. Bye bye.